Good afternoon and welcome back to the afternoon session. We'd like to thank the sponsors of our last break for so kindly sponsoring the meeting. Track 4, I'm going to be your host for the next 2 sessions, Science and Technology. My name is Pam Mace. I was diagnosed with a rare disease 16 years ago, and that's how I got involved and volunteer with Global Genes. This session is being live streamed, so we'd like to welcome all of our participants at home back. And our first session is New Approaches in Drug Development, RNA-Based Therapeutics, and it'll be presented by Dr. Marc Reidl, who's the Professor of Medicine and Clinical Director of the U.S. Angioedema Center at the University of California in San Diego. So please join me in welcoming Dr. Reidl. Thank you.
Well, thank you, Pam, and thanks very much for the opportunity to be part of this meeting. Congratulations to Global Genes for putting together such a wonderful event. And I was just sitting back there saying, I'm not even sure I'd go to my own session with all the cool stuff going on at the various times, but thanks for being here. I was asked to talk, as you heard, about new approaches in drug development, specifically about RNA-based therapeutics, which I think is certainly appropriate because there are a number of programs looking at rare conditions to see if this approach might be useful for controlling these conditions in a better sense. And so I'm at the US Angioedema Center at UCSD. We're about 3 miles from La Jolla Shores here, and I always like to start my presentations with a pretty picture, because then if you don't like the talk, at least you got to see a nice view. This is a nice place to spend a day. I sort of try to use this as a bonus to get my patients to come back and see us. You know, people get tired of going to the doctor, as you know, with rare conditions, and so I say, okay, well, just come to the beach and then swing by the office on your way back, and we'll talk. So I spend a fair amount of my time seeing people with rare immunologic conditions and swelling conditions, and so that's, of course, how I got involved in some of these therapies. I'm first and foremost a clinician, but do a lot of clinical research looking at new compounds, and I'll just finish my introduction here by saying I do work with some of the companies on a consulting basis or a research basis who are in this space. I don't think that'll influence what I'm telling you today, because I'm here for educational purposes, but be aware of those conflicts. So here's what I'd like to cover in the next 30 to 40 minutes. First, we'll talk about what is this approach? What is RNA-based therapy? What does that mean, and what does it involve? And then I'll talk about some of the potential advantages and challenges with this sort of therapy. And as we go along, I'm going to use some of the examples, some of the rare conditions that are being looked at for this therapy, so that you get an idea of how this actually plays out when we give it to human beings. What are we looking for, and what's the possible risks and benefits? So I was taught as a child that you're going to face a lot of tough things in life, and always start at the beginning. Okay, you got to start somewhere. So this may be review to most people in this room, but it's a fair question. What is RNA? And why are we targeting it? And so here's the simplistic view, is that RNA is the messenger. So you know the old saying, don't kill the messenger? Well, in fact, we're trying to kill the messenger here, but for beneficial purposes of treating a condition, a medical condition. And so as you all know, we have our DNA, which is sort of the sequence that's the building blocks of life. But DNA has a lot of its effects, of course, by encoding for proteins. It has a code that will build a protein, and proteins, of course, are the building blocks of our bodies and our physiologic functions. And RNA is absolutely critical. It's essential to get that message from the code over here to the protein that has the physiologic functions. Now, this is far too simplistic, this sort of drawing, because we know there may be up to 25,000 genes. We don't know the exact number, probably somewhere in 20 to 25,000 different genes in our DNA. But it turns out there's probably up to 1 million different proteins in our bodies. And so it's not a one-to-one ratio that there's one gene for one protein. And so you start to see the complexity that may be involved when we intervene on the system. So we know that there are genes that encode for multiple proteins, and they're modified in various ways, sort of epigenetic mechanisms, to sort of create the proper proteins at the proper time. But at any rate, this is the paradigm that's being used. If we can block this message, can we influence the course of a medical condition when there's abnormalities? And so just to put this in context, it's a little bit of a busy slide, but I think it sort of maps out how we're thinking about treating some of these rare conditions. So this is from a paper a few years ago, The Treatment of Genetic Disease. And if you look at those conditions that we know have a genetic basis, we know that there's a mutant gene, there's a problem in the code that's been either deleted or changed in some way. And because of that, you get this mutant RNA, messenger RNA, that's also abnormal because it's based on that code in the DNA. And then that mRNA actually leads to translation of a protein that's also abnormal. So the protein either isn't expressed correctly, or it's not folded correctly, or in some cases it's not made at all because the message doesn't properly translate to protein building. And then that protein, in many cases, leads to what we call the clinical phenotype, the symptoms that you get from the condition. And of course, that depends on what the gene defect is and the protein defect, but that leads to the symptoms that we define as an illness, a disease, a condition. So and I think this is right on. Again, it's complex, but this is basically the basis of genetic conditions as we understand them. Now, if we work backwards and say, okay, we want to treat this condition, there's different strategies that are used for that. And we start here at the bottom, because typically medicine has treated the symptoms. We've treated you have a condition, it causes the symptoms, well, let's treat the symptoms. That might be a pain medicine, it might be a medicine to bring down blood pressure, it might be a medicine to try to take the cholesterol level down if it's too high. But we're dealing with the symptoms rather than the root of the problem. As science has advanced and we got a better understanding of how this works, we're starting to swim upstream. And in some cases now, we can deal with a protein problem. So in people that have hemophilia or bleeding disorders, we might give back that protein so that we replenish what's missing. Or there are ways now to try to modify proteins, or monoclonal antibodies. Think of all the monoclonal antibodies that are out there now. They largely work by this. They're going to block a protein that's causing a problem and therefore modify the course of the illness. So that's pretty well established. There are lots of ways that we're doing this currently. But now we're going to take a step further, and we're going to try to get to the root of that protein problem, either with modifying the RNA signal so that the protein is either not produced if it's causing a problem, or I'll give you a very timely example in a moment of where you might modify the message so you actually make more of a protein that's missing. And that's one way. Or you're going to hear about this, I think there's a session after this, maybe we can even modify the gene. Maybe we can correct the gene, repair the gene, replace the gene. And so that's, of course, the whole field of gene therapy, which is also very exciting, but in its infancy. So this is just a way to think of how a disease progresses, and then also how do we treat that condition. And the purpose of this really was just to say, here's where we're going to be talking about for the next few minutes, is this square. Can we modify this message from the gene to the protein in a way that will improve human health? So this is just kind of presenting that concept. So when everything is working great, things are working perfectly, you have the gene that sits in the nucleus. It gets what's called transcribed, and you produce this green element, which is the mRNA or the RNA. And the RNA travels out of the nucleus. It goes to something called the ribosome, and the ribosome translates that messenger code into a protein. And then you have this protein that if it's green here, it's normal, and it's sort of working. It might work in the cell. It might go out of the cell and do things. But that's how this works. And then here's a different protein that's okay. And so you have this nice balance. Everything is sort of being transcripted and then translated in appropriate levels and working properly. So this is healthy. This is balance, if you will. Here's what happens in some of the conditions that we deal with where there's actually a problem. There's a disease-associated signal. And usually we think of this as a mutation in the DNA. So now you have these proteins, the red proteins, that are being either abnormal or being transcribed and translated at too high of a level. And so you can see, same process, but now we have far too many red proteins that will lead to a condition. And that might be, again, there's thousands of these conditions that are regulated by problems in protein translation. So too much transcription, too much translation, and you get this overexpression and an imbalance, and that leads to some symptom or some degeneration that we define as a chronic illness. So what we're talking about when we discuss the overall broad field of RNA-targeted therapeutics is really trying to restore this balance. Can we, through these molecules, and we're going to talk more about, they're often called antisense molecules, can we design molecules that will actually go and block these messages, block these red messages, so we got these blue molecules that are delivered to a cell, and they're going to basically match up and neutralize and or degrade these mRNA messages. So that at the end of the day, if we do it in the right amounts, the right doses, and of course this is where all the tricky details come in, can we restore some balance such that this red protein is no longer causing problems? It's knocked down or knocked out in a way that you have restored the balance. So it's this idea of neutralizing a signal, a messenger signal, mRNA, to restore balance. And you can see you still have a lot of transcription. You still get a lot of abnormal RNA being produced. To repair that, you probably have to go back to the gene, which again is another topic, but you can regulate the translation so there aren't these abnormal either levels or types of proteins floating around. So that's the goal. Now, this is a downregulation example. Again, the other way that's now being explored is, can you upregulate proteins? And that's the Duchenne story, actually, which I'll show you in a moment, is actually increasing the production of a protein that's deficient through this same strategy. All right, so we often sort of call these antisense molecules because they're actually trying to interfere with this message. And they're strictly defined as these molecules that contain a sequence which is complementary or has complementarity to a target RNA. So it's matched up so that it's like a magnet, basically neutralizes these RNA sequences. So these are designer molecules and therefore inhibits the function of that target RNA once it's bound. Neutralizes it, it can't carry the message, stops the protein translation. These are not necessarily new molecules. They've been described as far back as the late 1970s, but really it was in the 1990s that people started to realize there could be therapeutic benefits to using this if we could design specific molecules that were safe to use in this fashion. So it blocks protein translation. It may interfere with other RNA functions, and in fact, some of them actually degrade or destroy these RNA instead of just neutralizing them. And we can modify these. So the biological chemists can actually make designer drugs to improve the stability, the bioavailability, and the very specific binding. And this, of course, gets into the medicinal chemistry that's so important in pharmacology. However, since we're sort of messing around with these sequences, and again, these come from the normal nucleotide building blocks that we have in our own DNA and RNA. But since we're modifying these molecules now, we of course have to do a lot of safety investigations to make sure they're not toxic or they don't have unintended side effects from blocking some other message that we don't want to. So as always, we have to be very careful and study these closely. But the real message I want to get to here is that the hope of RNA-based therapy is specificity. It's this idea of a guided missile that you'll only hit sort of that one issue, that one message that's causing a problem. And of course, so you have to understand the science, the details behind a condition to do that. But nevertheless, I was trying to think of a good analogy for this, and I do not do social media, so this is a dangerous example. But I was thinking that if you had a Facebook account, which I don't, but if you had one, and you could design your Facebook account to ahead of time block all the annoying, irritating messages from people you don't like, that's kind of like this. You wouldn't have to think about it. You could block those messages before they came in, and you have to deal with the consequences or the angst of getting them. So it's this specificity of identifying a message, a protein that's causing a problem, and then designing a way to sort of preempt that production, to stop it before it causes problems. Okay. This is a busy slide, but to be thorough, I want to let you know there are various different molecules that will accomplish this. And so these are the four main types of antisense molecules, if you will. Usually that name has kind of been reserved for this oldest type, which are called oligodeoxynucleotides. But there's also small interfering RNA, DNazyme, and RNazyme. The details here aren't that important, but what it is important to realize is there are various ways to try to accomplish this. And the companies and the academicians that do this may select a specific strategy based on the disease they're dealing with and also some of the advantages and disadvantages of these approaches. So you can see they come in different chemical structures, they have different sizes. Some of them have enzymatic activity, so they will actually intrinsically or inherently degrade these messages, the RNA messages. Some of them don't have that, so they simply bind and then rely on cellular components to come in and actually degrade those messages. Some of them are easy to produce and get into the cells quite easily. Others are much more difficult because of the complexity and the size that's involved. So it's much harder to produce some of these molecules, particularly on the right side of this graph. And then there are always concerns about side effects. Some of these may have more off-target effects, meaning they might bind and cause some other problem in a cell or in the body. Some of them appear to be a little bit safer with regard to that. Some of them actually depend on the tissue that's involved because they require certain minerals or other chemicals to be present in order for them to work. And then finally, how stable are they? Some of them, you can make them, but as soon as you put them into the body, they sort of get degraded very quickly. So this is basically just to show there are lots and pros and cons, and it gets very complicated very quickly when you look at trying to do this. But the good news is there are options, and my guess is there will be more and more strategies. I'll tell you about at least one of those towards the end that is quite exciting. But different ways to accomplish the same thing of blocking this protein expression issue. And so this is all, of course, we talk about personalization in medicine a lot. I don't know if you can read this. It says, I'm worried healthcare has become too impersonal, doc. That's nonsense. Just relax and lay back on the barcode scanner, okay? So especially with rare conditions, but I think in all of medicine, we'd like to match a treatment to a person so that we can do as much as we need to do, but no more. And that's the goal of this RNA-targeted therapy. Can we give a treatment that will simply block this one message that's causing problems? Okay, so that's sort of the nuts and bolts of the background. Well, where does this actually apply in the real world? And you can see it's a pretty extensive list. I'm not going to talk about cancer, but there's a very large number of studies looking at various types of cancer. Can you interfere with the molecules that lead to cancer expression, growth, or even resistance to chemotherapy? Can you block messages that keep the cancer cells from being killed off by other agents? So this is a very exciting area that I'm not going to cover because I don't frankly know anything about it, but you can see where that might be helpful for cancers. Some of the things in my world that are not rare, but asthma. There are studies looking at these drugs to block signals that cause inflammation in the lung, causing asthma. Another non-rare condition, inflammatory bowel disease. So there are broad applications to this. There are actually infectious complications. This has been looked at for hepatitis C, for respiratory syncytial virus, even for HIV. And one of the strategies I'll show you at the end has been is sort of exciting potentially for HIV treatment. This one is starred because you'll see there, this is an FDA-approved indication. The first drug ever approved in this class was for CMV retinitis in certain people. It actually blocks the replication and the problems caused by CMV because it blocks the signal that the virus uses to replicate. So it can be used for infectious diseases. And then we get over here to some things that might be near or closer to our hearts in rare disease or rare condition treatment. So metabolic disorders. The second drug that was approved for this is for a type of hypercholesterolemia, and there continue to be ongoing programs that are pretty close in sort of late phase development for other types of genetic hypercholesterol disorders. I'll touch on the DMD story. You guys know this story was mentioned earlier today, and of course just in the last week or two, an approval, which is an RNA-targeted treatment for Duchenne. And then multiple sclerosis, some types of amyloidosis, and really the only reason I know anything about this is a condition I have some expertise in called hereditary angioedema, another rare genetic disorder. So you can see it ranges from those that are rare to those that are very common, but a lot of different applications. If we understand the protein expression that leads to the problem or even the genetic problem or the genetic mutation, there's a potential for using this RNA-targeted therapy. So let me walk you through a few examples here so that you can kind of see how this works in action. So this is homozygous familial hypercholesterolemia, HoFH. Some of you may be aware of this. This is a genetic hypercholesterolemia. It's very rare, about 1 in a million are the estimated prevalence. And these people have very, very high LDL levels, over 500 if they're not treated, which unfortunately leads to the development of very rapid onset of atherosclerotic disease, mainly heart problems. And you can see even during childhood, these kids will start to have heart attacks and cardiovascular issues because it's not good to have an LDL this high. It leads to plaques and cardiac events. And this has been traced back now to, there's a protein called ApoB100, which is really an essential receptor binding component for these lipoproteins, including LDL, not just LDL, but that's one of the most important ones for, that ApoB100 regulates. And so the idea here was that if you could target and basically reduce expression of ApoB100, could you reduce the LDL cholesterol and then subsequently modify the coronary risk that these kids and young people have? And the answer, it turns out, is yes, you can, and that's led to sort of an example of how this can work in real life and actually benefit people with this condition. So here you see the liver. I don't really know much about the liver, so I just show this up for effect. But a lot of the processing of cholesterol and a lot of the molecules that regulate cholesterol homeostasis come from the liver, of course. And so here's ApoB100, the bad guy in this case, who's overproduced due to the mutation and basically leads to lots and lots of, here it's VLDL, but also LDL being really elevated. So this drug called mipomersen migrates to the liver and actually, through that process I showed you, blocks the mRNA that lead to ApoB100 production and knocks down the level of this protein. And so there was a big trial, I don't know how well you can see this, but big trials showing different doses of this drug by a subcutaneous injection. And you can see here's placebo. Of course, we like to see nothing much happens with the placebo. That means you have a good chance to show an effect. And here's different doses of the medicine. And you can see at the 200 and 400 milligram doses, you see these dramatic drops in, this is ApoB100. So that's the protein we're interfering with here, and it works. You can stop the message and reduce the amount by about 30 to 40% a reduction in that protein expression in the bloodstream. And then here's the important one, LDL. Does it affect the LDL? Yeah, it does. They understood the condition correctly. And you see similar drops in that LDL, which we hope, of course, will translate to fewer heart attacks and fewer coronary artery issues in these people. So this is a great proof in concept, a drug that's now on the market being used based on this RNA targeting technology. And so, like I just showed you, this demonstrated these significant dose-dependent reductions in these various proteins that lead to problems. Now, as I tell my patients, there's no free lunch, right? There's always a price to pay for every gain that we have. So there were some side effects and some that weren't so bad, injection site reactions and flu-like symptoms. But there are some that are being watched closely. Because this molecule works in the liver, there have been some increased liver enzymes and actually some fatty liver development that's being tracked. Don't know why that is, and that's, I guess, one of the warnings about this type of technology is it's not always easy to predict what the side effects might be, but nevertheless, this has proven so far to be a fairly safe drug with proper monitoring. And so approved a few years ago. And I guess what I would say in addition is that because this worked, now it opens up the door to other possible targets. And here's some other proteins that, there's a list of them, but here's some other proteins that might be beneficial to target with the same technology that could lead to control of other cholesterol elements that cause disease. So that's an example, sort of a case proof, if you will, of how this can work. I'll talk for 3 minutes about something I actually know something about. Hereditary angioedema. So for those of you who don't know, this is a condition that causes recurrent angioedema symptoms that are very unpredictable. It's a genetic disorder, and these people have swelling of really anywhere on their skin. Their intestines can swell up and cause blockages, as shown by the ileum here, which is really blocked up compared to normal. And unfortunately, it can affect the face and the throat, that this can be fatal for some people because of this very rapid and severe swelling that closes off the airway. So we have some treatments now. We didn't have much until about 2009. And so because of that, we spent a lot of time poring over the cause. And this is not meant to put you to sleep, but this is the pathway that causes hereditary angioedema. These people are missing this protein. There's a mutation that causes them to be deficient in something called C1 inhibitor protein. And you can see C1 inhibitor has a number of functions with the red bars, but the most important is that it shuts off the production of bradykinin. This got shifted a little bit, but bradykinin is actually the chemical that causes the swelling, causes the blood vessels to leak, and the symptoms that I showed you pictures of. So if you're missing this protein, these other proteins crank out lots and lots of bradykinin and cause the swelling in an episodic fashion. So there have been many strategies looked at these different targets, but it turns out one of the good targets might be this protein called factor 12. If you can block factor 12, you basically turn off the faucet. You stop this whole process from being activated and ultimately leading to too much bradykinin. And so this is, again, trying to get at the root of the problem. Besides fixing the gene, which we're also interested in doing, can we look way upstream to the root of the problem where you have overactive factor 12 because you're missing the brakes on the system? Can you actually shut down the system and stop the swelling from happening? And it turns out that RNA-targeted treatments are a way to do that. So this has not gotten to humans yet. We're very close to doing that, hopefully. But these are two different groups that have presented data in my field. This is in mice, and this is in monkeys. So I will be the first to admit that what works in animals often does not work in human beings. But we are excited by this, that if you take a targeted construct that blocks the mRNA of factor 12, you can actually, even with a single dose, can knock down the production of that factor 12 for about 3 to 4 weeks. You can actually stop the expression or significantly reduce the expression of factor 12. And this is in monkeys, and here's some repeated dosing, but you can basically see the same thing, that you reduce the production of this by about 90% or so with these targeted RNA therapeutics. And so there's also some animal data to show that by doing this, you stop the leakiness, you stop the swelling that occurs. So remains to be seen whether this will work in my field, but the same theory, blocking this protein now that sits upstream from the problem and seeing if we can shut off the faucet and stop the symptoms from occurring. And then I do want to touch on, because this is timely, the Duchenne story. So as you all well know, this is a progressive myopathy. It occurs in boys because it's X-linked. And interestingly, I didn't know this until I did a little research. The dystrophin gene, which is the gene that has a problem in Duchenne's, is actually the largest gene in humans that's been identified. So it's a very complicated situation to deal with that based on the size of the gene. Most of these are caused by a deletion in one or more exons. About 2/3 of young boys have a deletion. And we know that if untreated, which there has been no treatment, that they start to have muscular problems early in life, and unfortunately have lots of problems as life goes on with respiratory insufficiency, cardiomyopathy, and unfortunately don't live beyond their 30s in most cases. So the idea with antisense therapy or RNA-targeted therapy here was, can you restore this dystrophin-encoding gene by correcting the mRNA mutation that causes this gene to not be produced, or this protein to not be produced? And I'm going to show you sort of how that works. But basically, there are ways by blocking mRNA, you can take away that stop sign. The mutation here has a stop sign that says, don't make any more of this protein. And so we're trying to bypass that so the protein can actually go on, be produced, and then lead to better muscle function. And then, of course, the overall goal is improve the course of the clinical disease. So this is a little complicated, but this is actually a little bit different than stopping protein production. Here you see the normal dystrophin protein. So if everything's working right, you make this big, long protein that's very important in muscle function. What happens in Duchenne is that in different exons, you can get a mutation that says, you see here, it says premature stop codon. That means that the message is being translated from the gene, and then it hits this exon, and because of the mutation, it gets a signal to stop. Don't make any more. Don't transcribe any further, okay? And so you get basically a partial protein that doesn't do anything. It doesn't do what it's supposed to. Now, the idea here is that if you block this, so here's a antisense molecule that's going to block that message in this exon 51, the one that's a problem in this particular example. And so by doing that, you're coding. We skip introns. Those don't get coded into proteins. And then you see 51, and it says, I'm not getting a message from here. I'll continue to go on to 52. So you basically cut out this dysfunctional exon that causes the stop message to occur. And what you get is a partial protein. Now, it doesn't look the same as this one. It's not normal, but it still has enough pieces to have some function and actually, we think, improve muscle function based on the studies that have been done. So this is the drug, eteplirsen, if I'm saying it correctly, as you know, just recently approved. These are fairly small molecules. They're a series of bases that have been modified to have this specific function. And so this has been a big step, of course, because this is the only treatment now that's been shown, we think, to modify the course. Now, I will say that one of the questions that the FDA was looking at closely, and I know it's been very controversial, so I'm not going to wade into the politics of the FDA, but what they were trying to determine was, does this drug do what they actually think it does, make more dystrophin? And one of the delays was to get more data on the muscle biopsies from these kids to see if you actually could see more dystrophin. Does it actually accomplish what the drug company says it does? And at the end of the day, my understanding was it was yes. They could actually see more dystrophin, this sort of partial protein that has function. And then the bigger question is clinically. That's a hard thing to measure in this group, but clinically, do you see improvements? So far, the answer seems to be yes, but I think over the next couple of years, they're going to have to look very closely to make sure it's actually making a difference. Is this protein production enough to make a difference in the course of the condition? Okay. So I like this quote. It turns out that medicine is hard, science is hard, and the universe is under no obligation to make sense to us all the time. So I want to touch on some of the challenges with this new area. One is, how do you give it to make sure that you get enough of these compounds where it's supposed to be to result in improvement? And it really has been looked in basically every way you can get something into the body. People have looked at that with these molecules. So topical or local administration, whether that's in the lung, is it in the nose, is it on the skin, in the GI tract, even into the eye or into the brain. These have been administered because you have to get enough into the local tissues to have an effect sometimes. But increasingly, we like the systemic route if it's safe, and so orally. Most of the drugs I've shown you are either given by sub-Q injection or intravenously, but there's other ways to do that. And so we think that these molecules are just taken up into the cell by something called endocytosis. They're sort of morphed into the cell and go into the nucleus. And so because of that, you don't have to, it doesn't appear you have to do a lot of preparation in many cases. They're so-called naked compounds. But there are ways now being looked at to improve the bioavailability or improve the delivery of the medication. So you can put it in a lipid nanoparticle that's more easily taken up into cells. You can actually put it into or link it to things that are targeted to specific meds. So if you want to, or organs, if you want to get it into the liver, if you want to get it into the brain, you can sort of link it to something that's taken up more readily by specific tissues. So these are sort of ways to fine-tune how we're giving the drugs. The other thing that's being looked at now, I alluded to earlier, is can you deliver these antisense molecule machinery to cells so that they could make these molecules themselves? Wouldn't that be cool? You could actually design tissues or instruct tissues to make these molecules and self-regulate the production of these proteins. That right now requires putting these, the antisense molecule machinery into vectors, viral vectors that go and infect cells, integrate into the genome, and then basically turn on machinery to crank out these antisense molecules. But this is what's been looked at or being considered for things like HIV, where you have a chronic infection, although it might apply to other chronic diseases, is sort of delivering the package, if you will, so that the cells themselves can crank out these anti-DNA molecules or anti-RNA molecules and block the messages we don't want to have around. So that's sort of, in the future, potentially, these are, of course, other ways to just get it more easily into the cell so it can do its job in blocking the proteins. So those are some of the known challenges, and then finally, the unknown challenges. Sometimes we don't even know what we're supposed to be afraid of, right? So that's why we do clinical trials, because we have to see if there are unexpected problems that come up. And so if you look at the potential adverse effects of this approach, as we start to design these molecules more specifically, they become more complex. We worry more about unintended side effects because we're messing with nature more and more, if you will. So those might be local issues, pain where you inject it or burning if you put it in your eye. But they could be systemic. I mentioned the liver toxicity that's been seen in one example. There have been some issues with bleeding or low platelets or long bleeding times in some programs. Maybe that antisense molecule, even though it's a designer to hit one RNA, maybe it starts to interact with other RNAs and it starts to silence other messages or genes that we don't want it to. So that's why we have to watch very carefully. Sometimes what you're having it carried in will have side effects. And then there's always a concern that when you're putting something into the body repeatedly, that you will cause an immune response that will cause more inflammation and more issues. So these are, drug reactions, or in this case, reactions to these blocking these RNA messages. So these are things, it's the importance of clinical trials, obviously, things we may or may not predict that we have to watch for. And I'll just finish by saying this is a hotbed of research right now. I did a PubMed search this week, and you can see that this is 4,500 publications a year we're up to now, and you can see that it's accelerated over the last 5 years or so. So over half of all the published work has just been in the last 5 years as we get more and more programs investigating this. And a lot of those, as I mentioned, are looking at better ways to design these molecules, and then are they going to be safe, which is, of course, of utmost importance. So I'll wrap up with this. This is a group in Denmark, actually, that tracks the developments in RNA-targeted therapeutics. The most recent I could find of this nice chart was 2013, so it's likely to be higher than this now. But as of 2013, there were over 100 different drugs based on this strategy in development. You can see most of them are either this interfering RNA or these antisense molecules. The first two columns I showed you in that chart, the sort of simpler and older version of things, but increasingly there's more complex ways of doing this being developed. And then this is just to show you that there are all these different flavors of how to do this. It gets very complicated, but most of these, again, are RNA or antisense, interfering RNA or antisense approaches, and most of them are quite simple, either naked or assumed naked, which I like that term. I'm not sure what it means, but they're not doing a lot, having to do a lot of processing to get this into cells and accomplish the targeting that they're after. So I'll wrap up with that. I'll be happy to take questions. I actually am from Missouri originally, so I'm a big Mark Twain fan. Most people don't know, he was actually a brilliant writer, but was also very interested in science. He used to hang around with Nikola Tesla and Thomas Edison, and so I think the goal is, of course, to help people, help patients, but I think Mark Twain would be very proud of these advances as well. So thanks for your attention, and I'm happy to take any questions you might have.
Dr. Reidl, thank you for that very interesting and educational presentation.
I've got two rather short, simple, but therefore open-ended questions. It just shows my level of understanding. You've talked quite a bit about the need for extensive testing for safety, and that's with any new technique, it's quite clear that people are concerned about that. Can you give us any idea of the relative safety factors with conventional drug therapeutics or drug treatment, since most of the factors seem to be the same, and there's the targeted nature of things like siRNA that are very appealing. It seems that you're not covering the whole pasture, you've just got that little segment by the fence. So that's my first question.
Yeah, I think so. It's a sort of a double-edged sword. With most of the drugs we deal with now, which are either small molecules or proteins, in the case of a lot of the antibody-based drugs, there's sort of more experience in dealing with that sort of technique, if you will. So you're blocking a chemical in the body, or you're blocking a specific protein. And so those drugs, of course, have lots of potential side effects too, especially the small molecules where we see lots of allergic reactions and other side effects because they're not targeted. So the good news is I think there's hope that this will have fewer of those types of side effects. The bad news is that this is sort of uncharted territory in some way, and that now we're getting closer to the root of the problem. And we're always worried about unintended consequences. It's sort of the best intentions can sometimes lead to other issues that we didn't expect. And a lot of that has to do with this affinity. Are we only blocking this one mRNA that we want to, or is it possible that we don't realize we're blocking some other message somewhere that we weren't even aware of could be a problem? And so it's the targeting is the plus, the sort of unknown is the minus, that we might have off-target effects that we're not aware of. I guess what's encouraging is that we do now have at least three drugs that have used this, that have gone through the human studies and didn't have a lot of those issues, except for perhaps the liver problems with the one drug. So the more of that we have, I think the more confident we'll be that we're not missing something important in using the strategy. But you just have to do, unfortunately, you have to do the studies and look at the data because some of these things may be quite unpredictable.
Hopefully we'll find that in 5 years' time it was much less of a concern than we feared.
Correct.
The second question is, we're going to go into a very detailed and, I'm sure, elucidating talk about CRISPR. Can you just, in broad terms, help me to understand, if we're talking about RNA treatment versus CRISPR, they're all new to us. How do I classify them? How do I understand the distinction between CRISPR, Cas9, and the RNA-based treatments?
Yeah, I would probably go back to this first slide because they're both trying to get at the root of the problem. But CRISPR, as you're going to hear about, deals with this box up here. It's going back to the DNA itself and seeing, can you repair or fix the problem at the root cause of the DNA sequence? And therefore, you will have a normal message that comes out of that DNA and a normal protein and sort of fix the problem at its root. This is not quite that far upstream. It's close. But here we're not actually dealing with fixing the actual mutation itself. We're just modifying the message that gets sent from that initial DNA.
So splicing or grafting of the RNA is not really put it in the same realm?
No, no. Yeah, that would be, DNA would be up here, yeah.
Thank you.
Sure.
Hello. My daughter was diagnosed with a very rare disease through whole exome sequencing. And what I understand from the immunologist we spoke to is that he doesn't know if there is an upregulation or downregulation. How do you test for that? How can you find out what you have to do with the mutant RNA?
Yeah, so this is one of the, I don't want to call it a trap, but it's one of the problems with exome sequencing right now, is that you can find through sequencing where there are abnormalities in the genetic code. What ends up being often more difficult is understanding what the consequences of that change are. So you have to know something about the gene. What do we think it does? What is its purpose? What does it code for? And then you have to make a link between that and the symptoms or the problem that we see clinically in the person. And so there's a lot of proteomics in between there. So what message comes from that gene? How is it changed in the RNA message? And then what's the problem for the protein that's encoded? So you have to know a lot of information in the middle there to sort of link between the mutation and the condition itself. And it turns out that's not easy, especially if you don't have a fairly large group of people to study. And as you all know, that's one of the real challenges with rare conditions is we don't have thousands and thousands of people that you can study to see where the problem lies. You're sometimes dealing with a few or dozens of people to do all that science in the middle from the code to the actual condition itself. So that's, I think exome sequencing has tremendous potential, but there's going to be a lot of hard work that goes into us understanding what these different mutations actually mean. And then once we have that, how to actually intervene. Yeah.
Okay, I think we have time for one more question. I think there's like 10 minutes between this session and the next one, but Dr. Reidl, would you be willing to take questions afterwards?
I'll be happy to stay afterwards.
Hashem, thank you.
So I represent an organization called the Alagille Syndrome Alliance, and Alagille syndrome is an autosomal dominant disorder. It's caused by two different genes. Jagged1 is the most common. And what happens is that it results in an insufficient amount of the jagged protein being produced. And within that gene, there are all these different mutations that are causing the disease. Does this type of therapy have potential for this type of disease?
So, good question. Depends. And again, this is where this is not going to be a panacea that's going to address everything. So the Duchenne again is a good example of that. As you probably know, the therapy that was just approved only is going to probably help about 13%, between 10 and 15% of the boys that have that, because it only addresses that one exon. The machinery is only going to help it hop over that one exon that's causing a problem. So you have lots of boys that have a different mutation where it's not going to do anything. It's skipping over the wrong thing. So the relevance might be that in the case of the condition you're talking about, where are the mutations, what problems do they cause, and then could you target sort of a way to bypass that mutation? Would you still get a protein that would have some of the important function and be helpful for the condition? So there's a lot of ifs, ands, or buts there, and it really is very condition specific based on the mutation type and the protein coding, whether this would be helpful to upregulate that.
Thank you.
All right, thank you for joining us today, and please join me in thanking Dr. Reidl for a great presentation.
Thank you.